Hey everyone, and welcome to Science in Real Time. This is the show where we sit down with the people pushing science forward and talk about what's really happening behind the breakthroughs. Before we dive in, make sure to subscribe so you never miss an episode. We've got some incredible conversations coming your way. All right, let's get into it. Scripps is really known for pushing the boundary of discovery. So I'm wondering, what makes it such a fertile environment for so?
Well, first of all, it's academic, okay? So everything we do, we really kind of want to publish on, as do our collaborators to a degree. So being part of Scripps and Scripps has international recognition, right? So folks will come to us with their, somewhat unexpected needs sometimes. But usually what we can do is engage very, very well and bring in their biology and interact it with our chemistry. And then build on it from there, and the whole drug discovery process from its very early onset, right, the beginning of the pipeline and small molecule discovery, all the way through medicinal chemistry and really preclinical validation can all happen right at Scripps. So we have that infrastructure in terms of automation and people and the skill sets and the chemists, pharmacologists. So it's a pretty unique atmosphere that way, which makes it very fertile.
How would you define high content screening today? Specifically, how has that definition differ or changed throughout the last few years?
High content screening formally became available from a vendor perspective in labs right around the beginning of the 2000s, all right? And they were very, very good sort of microscopes in a box that had unique features that would allow you to do high content analysis, not much different than what you see on the walls, right? With the floors and all that things that have now become more optimized. So the reagent tools are better, more appropriate, more accessible, more cost effective. But as the readers evolved, they were formerly very slow, all right? In order to get the resolution and imaging that you needed, it would take hours for even a 96-well plate with just a few fields of view and maybe at 10x objectives. These days, what's happened is really good engineering, really good science and physics came together, and you can effectively do it in minutes now. So multiple orders of magnitude faster than it used to be.
What kind of biological insight does imaging unlock that other readouts that are more traditional simply cannot?
A picture's worth 1,000 words, okay? And it really is. So in convincing myself and even scientists in my lab or folks that we're collaborating with, if I can put up an image that says this biology is interacting the most appropriate way it possibly can, and we're looking at molecular condensates or degraders or glues or all these things that pharmas and myself are very, very interested in solving problems around, cancer or neurological disorders or otherwise, you can now show that directly, right, with high resolution and very good confidence that you're doing the right thing. But what's happened now with the speed and the efficacy that I'm talking about, and obviously even bright field imaging can happen simultaneously. You can now look at images in minutes or seconds that used to take, dozens of minutes to hours. And now you can think about looking at kinetic modalities and how biology is changing in time. Whereas formerly, when it takes 2-3 hours to read a plate to get the information you need at the resolution, that's an endpoint type scenario, and it really limits your ability to look at the biology and the chemistry in sync with when you really should be looking at it. You think about neurite outgrowth and neuronal disorders, a very big problem, right? Alzheimer's, autism, etc. When we're screening for neuronal disorders, lots of times we're using either primary cells from mice, or now you can use iPSCs that are induced into neurons or neurospheres. And we do that very effectively in my lab, and there's lots of others as well. I would love to publish on that stuff. But what was happening is you would look at it at like day 3 in vitro after you added the compound, day 9 in total. Right? You weren't imaging over all that time. You had to have an endpoint because it's too slow. Fast forward now, it's like putting, I like to say, it's putting your goggles on underneath the water. You see things that are just quite beautiful early on, all the way through whatever you need, and it's very effective. And I think that's going to be very revolutionary in how we approach high content screening now.
Where do you see bottlenecks in high content screening workflows right now? Is it the biology, is it the instrumentation, or even how we analyze the data?
A lot of what's coming at us is the hardware usually supersedes the software. So you'll get really good engineering approaches then coupled to software that has to be refined and made more effective. And a lot of these cool ideas that the biologists and chemistry folks that have a biology problem will come to us with have to be, not only read on systems that are really fast and effective, but then you have to really learn how to deal with the data in a concerted way. And so when you're producing terabytes of data in literally minutes or hours, petabytes of data a day, which formerly didn't really exist so much, you have to find unique ways of dealing with that. How long do you store it? Where do you store it? You store it on the cloud? Do you store it on-prem in a server? Or, those types of things make a difference on how you access your data. Building in some principles around AI and dealing with, that data analysis in a hurry, all the while making sure that it's absolutely concordant with the outputs that you need to look at is going to be critical. And I think that's where the real build right now and the real energy is coming on the back end of what I call light speed type reading. So
Phenotypic screening has regained momentum in the last few years, and I'm wondering, what do you think is motivating this rediscovery?
First of all, to define phenotypic or phenotype in my mind means we're looking at cells and cellular responses. And there may be targets within the cells, for sure there are, because you wouldn't be screening them if they were, that you're interested in modulating, but you don't necessarily have to modulate them directly. You could have upstream or downstream interactors. And so the reports that you're looking at or the images that you're looking at are going to give you information around that, but it may not be directly on that target and you have to figure it out. First of all, I think one thing to remember is that most first-in-class drugs through high-throughput screening approaches arrive through high-throughput screening. So roughly 30, maybe a little bit more percent of all new first-in-class drugs are discovered through high-throughput screening. In this case, small molecule discovery. There's biologics and other things, large molecules that you want to think about as well in the same context. But that's where most first-in-class drugs go or come from. After that, fast follow-on approaches are more target-based. Screening and at capacities, at scales now where you can screen a full 1536 well plate in less than 5 minutes with 4 channels, all cells and all wells at 0.27 submicron level accuracy. You'll find things that we never expected to find in a hurry. And it allows you to get that throughput and the capacity that we never had. It was only things like luciferase reporters or HTRF results or things that are more target focused. Usually went at scale and at the speed that I'm talking about. So if you can achieve 10 plates an hour in screening, which we now can with the high content readers that are available, then we can certainly screen at scale, get those targets on and off our robots so that we can transition to the next target, all the while we're building in the secondary phenotypic deconvolution assays, right? Because you have to have the tools on the back side to solve those problems. We run those screens as well, and then out the back end comes something that's usually very specific, very on target, or you just go down other paths, and so that's how it goes.
Yes, that sounds incredible. And I'm wondering now, how would you say that high content imaging helps specifically teams embrace biological complexity rather than simplifying it away solely because they're able to see it?
I would say that they have tools now to look at all the images and all the cellular phenotypes that are arriving in a hurry versus when we didn't used to have that. So shorter answer, I think, is we're seeing things that we couldn't see before because we now have the tools to look at them faster and more appropriate time frame. I think it's going to change the way people build their assays, right, and configure them and use these high content approaches in real time, combined with AI models that will actually deconvolve the data better, allow us to understand how long we need to hang on those images for, right? Because storing data costs money, retrieving data costs money. This data, in this case, are images. You can do segmentation and all these other really cool things to try to deconvolve what your targets are doing. But you can also overlay and maybe even just use bright field to a large degree, AI imaging. And so you can look at phenotypes with controlled AI that will give you outputs that may happen very, very quickly. We're sort of at the forefront of that, I think, at least in the academic labs. I'm going to guess pharma may even be way out a little bit ahead of us, but at the same time frame, we all sort of converge on these ideas when you see the new tools. And so that's what we're trying to do. We're trying to exploit these new tools that will allow us to do things bigger, better, faster, more phenotypic relevant.
I will actually start thinking about that saying constantly, because bigger, better, faster is definitely how we want high content imaging to go. And I'm wondering, before we jump into AI and talking a little bit about automation and all of that good stuff, I was wondering, what does it take for phenotypic data, in your opinion, to be trusted alongside, or even more so, than target-based readouts?
Great question. I love that question because I often question data itself, right? You kind of have to. It's a a natural level of skepticism about what's happening and whether you're going to believe those results. So to me, and what I've actually done in our own lab with some of my postdocs and actually assay scientists, is we've run side by side some of these target-based approaches or even phenotypic cell-based approaches that have different reporters alongside imaging outputs that we can combine with AI. And those sort of outputs, now we can say, okay, well, here's what these results look like, and here's what these results look like. Are they concordant? What's the concordance of the data overlap, and how biased were we before, right? Because quite frankly, humans bias what we're doing, whereas these AI approaches that are unbiased will come up with a different set of configuration. So far, what I can tell you, and we're about to publish on this, is there is definitely some really strong concordance, but we're also seeing things that we didn't see before with AI, which I find very unique and interesting. We have to go down those paths very, very quickly. And we also find some things with these other types of approaches that the AI didn't see, right? And that may be more training the AI a little differently and all that. So there's all these iterations of events that quite frankly are speeding up and going faster because of the imaging and because of the AI. So I think we're going to get better and better at that. But yeah, I think you have to have sort of these, what I call head-to-head comparisons.
I'm definitely going to be looking forward to the paper, and we will definitely share it in Science in Real Time as well.
Yeah, it's going to be authored by Dr. Yuka Atsuka, one of my senior scientists, and it's coming out very, very soon.
AI is literally everywhere right now, specifically in imaging conversations. I'm wondering, what do you see as generally transformative over promise?
We're talking about imaging and drug discovery. You can think about that in the same way. We now have access to millions of data points and essentially billions of data points and information from around the world in a blistering hurry. And we can add to that in a very relevant way by really designing biology and sort of experimentation that approaches these questions, right? So I don't think the AI is overbaked in a lot of instances. I think it may be a little bit misunderstood, but I see real instances now where people are turning the switch. And quite frankly, I think if you don't get on board with AI, you're going to find yourself lagging way behind and maybe in a difficult situation. Potentially a really bad situation. Most big universities, including mine, have extremely powerful computing systems. In fact, we have the Hypergator at the University of Florida, which I think is arguably like the second most powerful computing system in the world, right? NVIDIA actually installed it. So of course we're accessing that, right? You don't have these powerful situations and not let them be used to their maximum. And we're learning those ways to engage them and sort of making, digital twins, if you will, of particular parts of our libraries or images, etc., and trying to deconvolve using, non-human but human relevant technologies, which is really AI, right? So
specifically, as somebody who works from academia, I'm wondering now about partnerships with folks that actually focus on AI automation and all that good stuff. So I'm wondering, how do partnership with technology developers, specifically like imaging and analysis platforms, can accelerate progress in ways that academia can't, and how that symbiotic relationship can help even more so?
As an academic professor and someone that tries to train and teach the next generation of scientists that are going to do what we need to be done, creating partnerships and actually being fairly extroverted about it and engaging very, very deeply is a critical component to success. So we can't build all the tools ourselves, right? We're limited in capacities and money and all these other things. But if you find the right smart people that are doing the right things and you actually make tangible, key points of handshakes, and you build upon those relationships successfully by actually doing what you're going to say you're doing, and they do as well, and then it's a convergence of great minds providing great tools. It's incredibly effective. And in fact, we're at the SLAS 2026, and that's sort of the birthplace of Tim Spicer, where I learned how to engage with, really industrial experts and tech providers that can do things that, I never thought of, or even if I thought of it, I couldn't figure out how to do it on my own cost effectively or in a time frame. And it's just building those components that you need. So imaging, right? You can use high content readers like the Araceli that will improve your speed and efficacy and really the precision and accuracy that you may need for your particular screening or others, but you may want to build on that, imaging AI, right? And now there's quite a few companies that are actually deconvolving fluorescence images and or bright field images in a blistering hurry. So all of these data outputs that are somewhat concerning from an IT perspective don't really have that sort of, scariness to them, because now you can combine them with good companies that are doing great things. There's some examples. Vicky AI is one example. There's there's also others.
At this point, I would like to go behind the science and talk a little bit about your leadership, what keeps you curious, and how do you proceed with collaborations. UF Scripps is a beautiful place to be able to be doing this type of science that is very needed. And I'm wondering at this point if there's any advice that you would give scientists who want to be able to pursue a career of the nature of yours.
I often say to folks that are coming into my lab to interview to join the lab, or maybe it's an intern and trying to coach them because they might be a little bit confused about a career path, set those tangible goals, but then when you're in this interview process, or you're even interviewing for school or to get accepted into the next level, you should be interviewing those folks as well, right? Because you kind of want to fit that mold, or maybe not be exactly that mold, but you kind of want to know that you're going to be comfortable in that situation, and therefore you'll be way more effective, right? Because work's not always easy, right? We tend to make it sound like it's great and fun all the time. It can be pretty hard, but at the same time frame, if you keep those goals in front of you and you're working in a good environment, something that fosters collaboration, fosters growth, you may get some negative feedback at times, and criticism is sometimes hard to take. But as long as it's in the appropriate dose and the right format, you can build upon individuals' careers, and that I think is kind of essential to think about as you're trying to grow into something like me. SLAS, absolutely a place, if you want to do a career like mine, is a place to engage all the time. So, for me, I think I mentioned it already, it was like a birth of Tim Spicer to become who I am today. And again, it wasn't just me, it was all these folks that affected me. So,
for listeners who want to stay in touch with you, or who would like to be able to engage with UF Scripps and do collaborations in the future, or simply are curious about what you folks are doing, where would be a good place for them to go and find you folks?
If you just search on Timothy Patrick Spicer, or Timothy P. Spicer, naturally you'll land on me. And then actually my email is spicert@ufl.edu. Happy to engage with you over email. You can find me on LinkedIn as well, again under the same name. Easy to collaborate that way too. Check out our website. It's scripps.hts@ufl, and you can see all the publications and papers. You can see the cool toys that we have. We have news and views, and we update that constantly, which is kind of cool. So I'm very proud of the fact that our engineers and Justin Shoemate in particular updates our website almost daily. So it's very relevant, very new, and up to speed. And yeah, definitely whatever your curiosity is, if it's high throughput screening from a pharmaceutical level, whether it's a new technology that overlaps with some of the work that we do and you want to introduce that, you're a student potentially, you want to do an internship, right? We always look at those folks too. Generally speaking, we have 2 to 4 interns every summer in my lab. My lab is 12 people, right? Including Louis Scampavia. Dr. Scampavia and I have been co-directing the screening center for about 13 years now. And yeah, so we always take in students. We both love to teach. In fact, we're teaching drug discovery and graduate studies right now at Scripps. Yeah. So we're still part of the Scripps Research Graduate Program. And Louis is back home right now. He's not here, or he'd probably be sitting right here next to me. He's teaching. So thank you for that, Louis. And it's just a wonderful, productive lab. So yeah, thanks for asking.
Absolutely. It is my pleasure. And all of that information will also be in the description box for people to be able to get in touch with you. Yep. And before we wrap up, I'm curious, is there one question you hope the next generation of scientists ask about imaging and discovery?
The curiosity in me is I don't really know how fast AI is going to come upon us. I feel like it's going to come very fast. And I also think that imaging and the veracity of imaging, how that's going to be controlled, right? There's a lot of ways to think about imaging. For me, I'm thinking about cells and biology, and maybe even higher level imaging, like you could look at mice and the effect on mice or whatever, how they might be in a certain new model, right? Because now instead of just putting them on a trapeze or whatever they call that thing, they actually use images to look at the way mice are neurologically affected by a particular drug or some disease. So I guess I'm going to be very curious and keep my eyes wide open on how fast this turns over from sort of conceptual AI into, really computational modeling of disease with really cool people that do that computational modeling and AI, and how that converges with biology. And so yeah, that's where I'm going to keep my eyes on and actually try to insert myself very heavily.
Absolutely. I feel like we all should be keeping an eye on that area in particular, and specifically for scientists that are coming into the field, keeping an eye on what is the latest and greatest and where are we moving, and having a part in shaping where are we moving as well. So that pretty much concludes our time together here in Science in Real Time. I want to thank you so much. This was such an amazing conversation. Being able to sit with somebody who has had such an amazing career and has been able to contribute so much to a field of this nature is a pleasure for me. This was an amazing experience, and I hope you enjoyed yourself as well.
I sure did. Thank you so much.
Of course, my pleasure. Thank you so much for tuning into Science in Real Time. We're really glad that you were able to spend time with us. If today's conversation sparked questions or ideas, we'd love for you to keep it going. You can connect with our guest and the Science in Real Time team using the links in the description box below. And if you enjoyed this episode, don't forget to like, subscribe, and share with someone who loves science as much as you do. Until next time, thanks for watching!